On behalf of the Biochemical Society and Portland Press, I'm pleased to welcome you all to today's webinar, which is part of our Biochemistry Focus webinar series. Topics in this series include different research areas in molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please go to our website for more details. Myself, Kakoli Bose, PI, working in the area of biophysics and structural biology at the Cancer Research Institute at Utrecht Tata Memorial Center, Mumbai, India, which is one of the premier research institutes and the largest cancer hospital in Asia. My research involves understanding protein-protein interactions in the cell death pathway, also known as apoptosis, and their implications in cancer and associated diseases. I'm also an editorial board member of Biochemical Journal and associate editor of Bioscience Reports Portland Press. In this webinar, we will hear from the Biochemical Society's 2022 AstraZeneca Award winner, Dr. Sjors Scheres, at the MRC Laboratory of Molecular Biology, UK. Dr. Sjors studied chemistry at Utrecht University, the Netherlands, where he also obtained his PhD in protein crystallography. He was a postdoc in the group of Jose-Maria Carazo in Madrid before he started his group at the LMB in 2010. Since 2018, Sjors is also joint head of the Structural Studies Division. His main interests lie in the development of new methods for high-resolution cryo-EM, structure determination, and their application to amyloid filaments. During his award lecture, Dr. Sjors will discuss the latest developments in cryo-EM and their application to amyloids from human brain. If you have any questions, start typing in the text box while the lecture is going on. Now I welcome Dr. Sjors, and my heartiest congratulations to him for this tremendous achievement, and I would invite him to start his lecture. Thank you all.
Thank you very much, Dr. Bose. Can you see my slide all right?
Yeah, I can see your slides.
Thank you very much, and also thanks very much to the Biochemical Society for recognizing our research with this fantastic award. It's a true honor to receive it. And at this point, I'd also like to thank all my past and present group members and collaborators who have all contributed to the research that led up to this award. So I'd like to speak to you today about recent developments in cryo-electron microscopy, and then also about our work on applying those developments to determine structures of amyloids from human brain. What you see in this rather gray slide is a picture that was taken in one of our electron microscopes, and it's a sample that was made by Israel Fernandez, a postdoc in the Ramakrishnan lab here at the LMB, containing bacterial ribosomes. Each of the little darker dots that you see is a individual ribosome complex, and these ribosomes were quickly frozen in a thin layer of buffer and fast enough in liquid ethane so that you get a vitreous form of solid water, which is non-crystalline. You then put this inside the electron microscope, and you take this transmission electron microscopy image. And each of these little black dots is a ribosome, but because these ribosomes were tumbling around randomly in the solution at the moment of freezing, each of these little sub-images represent 2D projections of the ribosome in different orientations. Now, in cryo-electron microscopy structure determination, we combine all these sub-images of each of these individual ribosomes into one three-dimensional reconstruction. To explain how that works, I find the projection slice theorem to be a useful insight. So if there is a three-dimensional object, then in the computer I can calculate its three-dimensional Fourier transform. This is just a mathematical operation, and it expresses the same information in a different manner. Now, if in the electron microscope I would take a projection image, for example, along the red arrows that are shown here at the top of the slide, I get the two-dimensional projection image on the right here, and this two-dimensional projection image, I can take a two-dimensional Fourier transform using the same mathematical operation that I did on the 3D object here. Now, the projection slice theorem says that if this two-dimensional image is a projection image of this three-dimensional object on the top left here, then at the bottom right, this two-dimensional Fourier transform is actually a central slice through the three-dimensional Fourier transform on the bottom left of the original 3D object. This means that if I know all the relative orientation, and the orientation of the 2D slice is perpendicular to the direction of the arrows in the that the original projection was taken by. Now that means that if I would know all the relative orientations of many two-dimensional projection images in different orientations, I could then put many differently oriented 2D slices into this three-dimensional Fourier transform, thereby filling up the entire three-dimensional Fourier space. And I can then just do an inverse Fourier transform and get a three-dimensional reconstruction. And that, in a nutshell, is what happens in cryo-EM single particle structure determination. Now, my group at the LNB focuses on two different topics. On the one hand, we develop methods to do the cryo-EM image processing that is involved. We implement these in the program called RELAN. On the other hand, we apply these methods to a particular system of our own interest, which is amyloids, and we work on proteins tau, amyloid beta, alpha-synuclein, and others. I'll first speak about the image processing part. So RELAN, which stands for Regularized Likelihood Optimization, is an open source software project which aims to do the entire cryo-EM structure determination pipeline from the raw images taken at the microscope to a final reconstruction of the three-dimensional electron scattering potential. We do this proudly open source because we're strong believers that doing things openly in science, also in software development, is ultimately best for the progress of science. And it is this software package that determines all these relative orientations, for example, of the ribosome particles that I showed in the first gray image. So determining all the relative orientations then allows us to put back all these two-dimensional Fourier slices in the 3D transform, do the inverse transform to get our reconstruction. Now, RELAN became available to the community in 2012, and not much after, around 2013, the field on the cryo-EM reconstruction field underwent rapid progress in the resolution that could be obtained from cryo-electron microscopy reconstructions. To illustrate that, this slide shows with black points these are all entries from the electron microscopy database of structures done in the 21st century between the year 2000 and 2010. And you can see that resolutions were mostly in the red zone here, up to, let's say, 10 angstroms resolution. And then in favorable cases, some of the resolutions went to slightly higher resolutions in the orange zone here. There's somewhat of a trend that higher molecular weight gives somewhat higher resolutions, and that is because there is more signal in the images if the molecules are larger. However, for very few structures, it was possible to go beyond 5 angstroms and reaching resolutions where you can do de novo atomic modeling in the map, which require around 3.5 to 3 angstroms resolution, were mostly absent. And this changed in 2013, and the red dots here are all structures that were determined that year. And the reason why suddenly there was such a big jump in resolution from all these structures where de novo modeling was not possible to a lot of structures where it was possible over a wide range of molecular weights is twofold. One was the development of better cryo-EM image processing procedures, for example, in Renown, and the other was the availability of new electron detectors, which gave images with a higher signal-to-noise ratio. Now, our part in that was the cryo-EM image processing pipelines. And one thing, which I won't go too much into detail, but what has been very important for the field, was the possibility to circumvent elaborate sample purification methods for structure determination. Because in conventional structure determination techniques, if you would have a mixture of different structural states in your tube, you would have to run biochemical purification methods to separate these into homogeneous preps on which you would then, for example, do X-ray crystallography or so. Now, cryo-electron microscopy is capable of circumventing this need for purification of pure samples, and that is because we can take images of mixtures. And the same program that is used to align to find all the relative orientations of the different particles can also be used to classify different structural states into different subsets of particles. And by doing so, in the computer, we can then take images of mixtures and process these images and get for example, two structures of both the red and the blue state in this example out simultaneously. And this has become very important part of structure determination, where now people can do mixtures of all kinds of functional states all together and get multiple structures from a single sample. I thought what I'd focus more on today was more recent developments, where we collaborated with Abhay Kotecha at Thermo Fisher factory of the electron microscopes in Eindhoven. And there we had access to a prototype microscope with a cold field emission gun to generate electrons that are more monochromatic, a new energy filter which filters away some of the electrons that have lost some energy in the samples and generate noise in the images, and a next generation of these direct electron detectors called the Falcon 4. And it was the combination of these three new elements of hardware with our software and a beautiful sample of a test protein called apoferritin that we were then able to calculate a 1.2 angstrom resolution cryo-electron microscopy map for the apoferritin molecule. So at 1.2 angstroms resolution, because the distance between non-hydrogen atoms within a protein is mostly above that distance, you can see for almost all the non-hydrogen atoms in the map, you can see individual blobs of density. So for example, here you see a phenylalanine with the 6 carbon atoms in the ring in the middle of the image. Now, the hydrogen atoms are not visible directly in this map because their intensity of the scattering potential is much lower. But if you now what we found together with Garit Murshidov, the author of RAFMAC, if we make difference maps and we subtract models which have only the non-hydrogen atoms from the cryo-EM map, then in the difference map we can actually see individual peaks not only for the non-hydrogen atoms, but now also for the hydrogen atoms. And right here at the bottom, you can see my favorite molecule in this entire map is not apoferritin, but this is one molecule of water. We have two green blobs for the hydrogens and one big blue blob for the oxygen, H2O. Now, in the remainder of my talk, I will speak about an application of cryo-EM structure determination that was made possible by another methods developments in the lab, and this was done by Xiaoda He, a PhD student, who implemented helical symmetry inside our software package Renown. And this made it then possible to look at filamentous proteins using our software. And this then spurred on a collaboration with Michel Goddard, who is a group leader also at the LMB in the neurobiology division. And together we've worked for about 6 years now on multiple neurodegenerative diseases. And one family of neurodegenerative diseases we've been particularly interested in is the family of the tauopathies. These are about 20-odd different neurodegenerative diseases that are all characterized by the filamentous aggregation of the protein tau, hence their name tauopathies. Alzheimer's disease, here indicated with AD, is the most common tauopathy, but as that there are more than 20 members of this family. And at the histology level, what you see here is immunohistochemistry staining of brain sections of individuals who have died with different tauopathies. You can see at the cellular level already these diseases look all very different, even though this is the accumulation of the same protein tau to form these filaments. Now, tau in its normal happy life is a monomeric protein. It's mostly unstructured and it binds to microtubules where it's thought to stabilize microtubules. And Liz Kellogg in Eva Nogales' lab in Berkeley did a cryo-electron microscopy reconstruction of microtubules with tau constructs bound to them, and which has yielded insights in how tau may stabilize these microtubules. But then somehow in disease, the tau dissociates from the microtubules, and then the monomers self-assemble into very long filamentous helices, which you can see a picture of here on the right. Now, if you would put a preparation of these filaments in an X-ray beam, you get this typical X-ray cross beta diffraction pattern, where there's a very strong signal at 4.7 angstroms and a weaker signal at a perpendicular angle at about 10 angstroms. These are caused by the observation that the individual monomeric molecules of tau assemble into very long twisted beta sheets along the helical axis, and there you get the typical inter-beta strand distance of 4.7 angstrom, which results in the 4.7 angstrom peak of the X-ray spectrum. And then these beta sheets, they fold against each other with typical cross-beta packing distance of about 10 to 12 angstroms resolution, which gives rise to the perpendicular direction signal there. Now, not all of the 441 amino acids of tau form these beta sheets. It's only a part of the sequence that forms these beta sheets in the filaments. And then the rest, both at the N and the C terminus of this so-called ordered core, flops around randomly. And this was already observed by Tony Crowther when he looked at negative stain images of these tau filaments derived from the brains of Alzheimer patients in the 90s, and Tony termed these random termini the fuzzy code. Now, in human brain, there's 6 different isoforms of tau expressed, and by the splicing out of exon 10, which codes for one of the 4 microtubule binding repeat regions of the tau sequence, you can then get either 3 isoforms which have all 4 microtubule binding repeats, and we call these 4 repeat tau isoforms, or by splicing out exon 10, you then get 3 isoforms which only have 3 microtubule binding repeats, and these are the 3 repeat tau isoforms. Now, people have looked at tau filaments extracted from brains of individuals who've died with different tauopathies, and have found that in Alzheimer's disease or chronic traumatic encephalopathy, for example, you have a mixture of both 3 repeat and 4 repeat tau isoforms in the tau filaments. But in other tauopathies, like in Pick's disease, you only have 3 repeat tau isoform aggregating, and in multiple other diseases like cortical basal degeneration or progressive supranuclear palsy, you get only accumulation of the 4 repeat tau isoforms. Now, this combined with the observation that the aggregation of tau seems to spread throughout the brain as disease progresses, has then led to a model where tau might behave in a prion-like manner during the advancement of the neurodegenerative disease, where assembled filaments of tau can then convince monomeric, otherwise happily disordered tau to join in and form even bigger aggregates, which then could split into multiple smaller parts, and you get an exponential spread. And that's kind of the underlying hypothesis of prion-like spreading in prion diseases. So tau could do the same here, and it could be that different prions, what is called a strain in the prion field, or different conformations of these assembled forms of tau, could be responsible for the different diseases, ultimately leading to these different disease characterizations, not only at a symptom level, but also at the cellular level, as I showed you before. Now, how that would work was very much unclear when we started this work at a structural level. So with the advances in the cryo-EM detectors and the software, also with the availability of Sjors helical reconstruction algorithms, we thought it would be a good time to apply cryo-EM structure determination to this problem. So we started with the brain of a 74-year-old lady who had died with advanced stages of Alzheimer's disease, and we isolated using Sarkisil solubilization followed by ultracentrifugation techniques. You can isolate tau filaments from the brain, and this was already performed back in the 80s and the 90s by Michel Goddard and Tony Crowther as well. So, but now we could take images of these tau filaments in our new cryo-electron microscope with the new detectors and use our helical reconstruction software to reconstruct these structures. And this here is an image of that preparation, and with the blue arrows you can see the type of filaments that Tony had already observed using negative SANE in the 90s, called a paired helical filament, which is characterized by a narrow wide variation. And then with the green arrow you can see a short, what Tony called a straight filament, because it lacks this variation in width. Now, using Sjors software, then Anthony Fitzpatrick, a postdoc in my lab, was able to reconstruct both the paired helical filament and the straight filament to resolutions of around 3.5 angstroms, which is enough for de novo atomic modeling. And here, false colored on the left in blue is a paired helical filament, and in green a straight filament in the micrographs. And then if you would zoom in and remove some of the noise through averaging, you could see this typical repeat of 4.7 angstroms, which is the distance between individual beta strands of the beta sheets. And then here in blue and green in the middle is the actual three-dimensional reconstructed density. And if you turn this on its head, so not looking along the helical axis, but now down the helical axis, you can see these what we call XY cross sections on the right, again in blue and green for the paired helical and the straight filaments. And you can see that both these filaments are made up of two individual protofilaments, which are C-shaped. And in the paired helical filament, the C-shaped filament pack against each other in a symmetrical manner. And this structure is much wider than it is high, and that's the reason why projections of these filaments have this typical variation of wide and narrow width. The straight filament at the bottom, the two C's come together in an asymmetrical manner, and the structure is more or less as high as it is wide, and hence the absence of this variation of width in the two-dimensional projection. Now, as I said, the resolution of these maps was sufficient for de novo atomic modeling. So Anthony found out that the ordered core of the C-shaped protofilament was made by the residues valine 306 all the way up to phenylalanine 378, which means that there is up to 105 residues flopping out randomly in the fuzzy code on the N-terminal side of the structure, and 70-odd structures on residues are part of the fuzzy code on the C-terminal side. And then there's 2 molecules of tau for each of these layers of the beta sheet in the filament, because you have 2 C-shaped protofilament, each containing 1 molecule of tau per layer for the paired helical filament. Now, the straight filament is made of the same 2 C-shaped protofilaments, again made both of valine 306 to 378, but they now come together in an asymmetrical manner. And the packing seems to be mediated by these 4 lysines, which all point towards a density in between the side chains of the 4 lysines, which we hypothesize is some sort of a polyanionic molecule which kind of neutralizes the charges of the positive side chain of the lysines. But to date, we still don't know what molecules cause this density. Now you can overlay the atomic models of the paired helical and the straight filaments, protofilaments, and you can see that the structures are more or less the same, which makes these filaments ultrastructural polymorphs. Now that was the brain of one individual with Alzheimer's disease. Would the next individual with Alzheimer's disease have the same filaments? To answer that question, Ben Falcon and Wenjun, a postdoc in Michel's lab and my lab, they worked together and looked at another 15 Alzheimer's disease cases with negative stain electron microscopy, and they did an additional 3 cryo-electron microscopy structures of 3 different cases. Sorry. And what we found is that in all cases of AD, you have present both paired helical filament and straight filaments, and it tends to be that there is more paired helical filaments than straight filaments, although the ratios vary a bit from case to case. But we now think that these paired helical filaments and the straight filament are characteristic of Alzheimer's disease. So if you have an individual with Alzheimer's disease, they will have these filaments in their brain. Now, the extent of the ordered core then explains why you get a mixture of both 3 and 4 repeat tau isoforms in the filaments. That is because the ordered core starts at valine 306, which means that the entirety of the second microtubule binding repeat, which is the one that gets spliced out with the 3 repeat tau isoforms, is absent from the ordered core. So it doesn't really matter whether R2, here indicated in orange schematically, is present or not, because this is all part of the fuzzy code where the tau molecule is disordered. So you can get an indiscriminate incorporation of both 3 and 4 repeat tau isoforms in the structure. So how about a different tauopathy that also has a 3 and 4 repeat tau isoforms in the filaments? And to answer that question, we looked at chronic traumatic encephalopathy, which is a tauopathy that some people develop after repetitive brain trauma. So in the US, it's famous for, there's some famous cases of ex-professional boxers or ex-professional football players. But in this country also, the normal English football players, there have been some high-profile cases where headers are thought to have been a cause of it. Now, it turned out we got access to CTE brain samples, and we could image tau filaments from these. And again, we used cryo-EM image processing developments to further our research on the application side. So using a newer version of our software, we were able to go to resolutions now well beyond the 3.5 angstroms that we had obtained for Alzheimer's disease, now going to 2.3 angstroms, thanks to developments by Jasenko Zivanov, a postdoc in my lab, who was able to correct for misalignments of the microscope a posteriori after the data have been acquired, and correct for it, and thereby get much higher resolution. And using these high resolution maps, we could then look at the structures from CTE filaments of 3 different cases of CTE. Case 1 was an ex-professional American football player, and cases 2 and 3 were ex-professional boxers. And again, the different cases all have the same structures. And again, there's 2 types. We call them CTE type 1 on the left, and then cases 1 and 2 had a second type, which we called CTE type 2, which was a minority, which we could not detect for case 3. And the structures are again C-shaped like, but the C is more open, and the way they pack against each other is also different from the AD fold. But even the actual protofilament fold is slightly different. So as I said, it's more open. And what we saw was the presence of an additional density. If you look here in case 1, it's most clear here at the tip of the C. This density was not present in the AD fold, and that's because when the whole structure was opened up, the C is more open than for the Alzheimer fold. Also, there's a bigger cavity between these beta strands 4, 5, and 6, which is now filled with this density. And because of the hydrophobic nature of this cavity, we had hypothesized that this cavity may be filled with hydrophobic molecules. Again, just like the molecule that seemed to hold together the straight filaments in Alzheimer's disease, we do not yet know the identity of these molecules, although near the end I will make a speculation as to what they might be. Now, since then, Michelle and I have continued this work, and we've solved structures of most of the most important or the most common tauopathies that are out there. And as you can see here, these structures are all different. So different diseases may have different faults. And whereas the Alzheimer's disease and the CTE-fold were relatively simple, it was both C-shaped, which was just more opened up. For example, in Pick's disease, you get a completely different elongated J-shaped fold. And you again get very different folds for corticobasal degeneration or here PSP. And some diseases resemble each other more than others, which led us to propose this structure-based classification of tauopathies. And even we were able to identify a new disease by doing the structure of a tau filaments from an individual that was originally identified to have diagnosed with PSP, but in hindsight had characteristics that were not in line with PSP, and their structure was actually different, suggesting that this individual suffered from a different disease. So for all these diseases, we did multiple structures, and individuals with the same disease always had the same tau filament folds, but as you can see, among the different diseases, the folds are very different. And as was true for Alzheimer's disease and CTE, that the protofilament, the tau isoform composition of the filaments, having both 3 and 4 repeat isoforms, was explained by the extent of the ordered core. The same is true for the 3R tauopathy Pick's disease, where R1, because R2 is spliced away now, is directly part of the ordered core, and the same for the 4 repeat tauopathy, where in this case R2 here in blue is part of the ordered core. Now, as a structural biologist, this kind of continues to amaze me, how you have one amino acid sequence for one protein giving all these different amyloid structures. This is very different from the globular folds that nature has converged on with evolution, where one protein sequence leads to one globular fold of a protein, perhaps with distinct conformational states, but still there is a correlation between protein sequence and a given protein fold. This does not seem to hold for amyloids, where the same protein can adopt many different structures. And yet these different folds then characterize very strictly different diseases. So multiple individuals with the same disease have the same structures, but different diseases and different folds characterize different diseases. So where the specificity of the different structures in the different diseases comes from is completely unknown. So this, I think, are important questions for future research, is what leads to the formation of these different structures in the different diseases. Do cofactors, for example, play a role? And I've pointed out a few of these additional densities in the straight filaments or in the CTE filaments, but we see similar additional densities in some of the other filaments from the different tauopathies. Are these perhaps post-translational modifications that remain to be identified? Or do truncations of tau or the actual isoform composition of the available tau molecules in the particular cell where this happens play a role? To answer some of these questions, I think we need better model systems, and one way to study this would be to develop better in vitro aggregation model systems, where we can control the tau protein that we put in, and we can control the factors, the molecules that are present in the reaction mixture. Hopefully, knowledge gained in such in vitro experiments could then be expanded to cellular models and perhaps even animal models, but that would lie further in the future. Now, a popular system to form tau filaments in vitro is through the addition of heparin. This is because full-length recombinant tau made in E. coli is extremely soluble and very hard to get out of solution to form filaments. Yet if you add small amounts of heparin, then recombinant full-length tau spontaneously assembles rapidly into filamentous structures. And Wenjon in my lab then did cryo-EM of both full-length 4-repeat and 3-repeat tau from E. coli with the addition of heparin and solved their structures, which are shown schematically here. And as you can see, these structures are very different from the structures that I just shown you for all the different diseases. So heparin-induced tau filament formation is not a good model system if you want to study tau aggregation and disease, because the outcome is a different set of structures. Now, it was only through the hard work of a PhD student in my lab, Sophia Löwestam, that last year we managed to make tau filaments from recombinant tau expressed in E. coli in a test tube that were the same as those from Alzheimer's disease. And the trick that Sophia had to do was to use shorter constructs. So instead of using full-length tau, she had to truncate tau, and she started with a construct that was described by that was discovered by the group of Louise Serpol using tau 297 to 391. Using the conditions described by Louise, she then found structures which again did not resemble Alzheimer's disease filament. But by further tweaking the conditions, the shape, the amount of shaking, and the addition of certain cofactors like dextran sulfate or magnesium chloride, Sophia was then able to make tau paired helical filaments with the exact same structure as those observed in disease. And you can see an overlay of her in vitro assembled structure backbone in gray and the AD structure at the bottom right of the slide here. Now, while doing this, Sophia scanned all types of the addition of all types of molecules. And one thing she noticed is that if she added sodium chloride to the buffer, then the same construct of tau would assemble forming a CTE type 2 filaments, so the minority type of CTE filament, but again with the more open C-shaped structure, and again with this additional density in the tip of the C-shaped protofilaments. Now, if she would change the cation of the salt that she would add to the buffer, for example, using lithium chloride, potassium chloride, then she could see that depending on which salt she added, the opening up of the C-shaped protofilament, the amount of opening up shown here at the middle bottom, it varied with the ion, and going all the way from magnesium chloride being more closed in the blue structure here with that is the same as the Alzheimer's disease structure, to the structure with sodium chloride, which is in orange here, which is the one with the CTE. And together with this opening up of the whole C, she could see that the extent of the cavity of the tip of the C inside would change as well. Now, especially the structure here with potassium chloride, where 3 protofilaments come together to form this kind of Mercedes star-like beautiful structure. For that structure, she got relatively high resolution, again using the same prototype electron microscope in Eindhoven. And at, I think it's 1.8 or 1.9 angstroms resolution, she could see that the additional density in the tip of the C was actually separated into 2 individual spherical blobs of density that for each rung of the beta sheet. And she could model these densities with a potassium and a chloride ion, and the geometry of the whole of the arrangement is consistent with what could form a potassium chloride structure. So what we think is present in the tip of the C of this potassium chloride structure is actual potassium and chloride ions. Now, I told you the in vitro assembled structure with sodium chloride looks very much like the CTE type 2 structure, and Sophia didn't manage to get that structure to that high resolution, but I think at a resolution of around 2.3 or 2.4 angstroms, she could see a separation of the density along the beta rungs, but not into individual blobs of density. but still she could model these blobs with pairs of sodium and chloride ions. So what we think is present in the tip of these C-shaped protofilaments in the in vitro assembled filaments are sodium chloride and potassium chloride ions. And that this is the in vitro assembled in panel G. Here's the in vitro assembled structure from SOPHIA. And in panel H is the same view, the CTE structure that we've done from the American football player. You can see the density in the channel is different. Even though they're at similar resolution, the density for the CTE structure is not separated along the beta runs, where it is for the sodium chloride structure. And so we don't know whether in CTE it's also sodium chloride that assembles into this channel, or whether again it's another type of molecule or salts or whatever. We don't really know, but it could be, given that the rest of the structure of tau is exactly the same, that in CTE sodium chloride does play a role. Now, while doing this, while scanning through different construct lengths and the addition of multiple different factors, looking at different pHs, etc., shaking speeds, SOPHIA in total, using all her recombinant tau made in E. coli, solved over 76 structures, which all went in one paper. And also shown in this slide are all different structures that we hadn't observed before. So what I've shown you before, that the same protein can adopt multiple different amyloid structures, is not only true for these different tauopathies, for the different diseases, but even just starting from E. coli expressed tau, there is a wide variety of different structures that the same protein can adopt. So that brings me to my conclusion slide. I've shown you that atomic resolution cryo-electron microscopy is now a possibility, and I guess you will be fully aware of this now. There is a load of structures, there's many structures coming out, and the opportunities to classify structurally heterogeneous data sets into multiple structures is very powerful in many realms of structural biology. And in terms of our own application of these methods to tau amyloids, I've shown you how different folds of tau characterize these different tauopathies, and yet in vitro assembly of tau, which we would really like to have as a model system to study how this all happens, may yield yet very different structures once again. And SOPHIA's 76 different structures are an extreme example of that. But fortunately, we are now able to make the Alzheimer's disease and CTE structures in vitro, which opens the road to start to do kind of functional assays in, for example, cellular seeding experiments, where you can explicitly look at structures where the only difference is the conformation of the tau filaments. Now, as in general about amyloids, I think we're learning that amyloid structures are much more structurally versatile than the globular protein domains, as I mentioned before. And we've now abundantly shown this for tau, but we're also seeing similar results for other proteins. In our own lab, we've looked at amyloid beta filaments from Alzheimer's disease and other diseases. And Ben Falcott's lab here at the LMB has looked at TDP43 from different types of diseases. Together with Michel, we've also worked on alpha-synuclein, which forms filaments in a different family of neurodegenerative diseases, which are called synucleinopathies. And Parkinson's disease is the most common synucleinopathy. And previously, we had already determined the structure of alpha-synuclein filaments from a different synucleinopathy called multiple system atrophy. And we found 2 different types of filaments made of 4 different types of protofilament structures. And very recently, which is now out on bioRxiv since a month or so, we discovered the structure of the alpha-synuclein filaments from Parkinson's disease, where we see that alpha-synuclein filaments are made up of a single protofilament, again with a different protofilament structure than those observed in multiple system atrophy. So what held for tau is probably also true for alpha-synuclein, where different folds characterize different diseases. And then interesting, we also looked at dementia with Lewy bodies, which and Parkinson's disease dementia, they all share the Lewy fold. So as is the case in tau, there is certain members of tauopathy family which have share the same structure of the protofilaments. This is also true for alpha-synuclein. And in vitro attempts to make these structures of alpha-synuclein as we observe them in human brain would now be necessary to kind of go the same way as we did with tau to get better model systems of forming these systems in vitro. So that's the direction we will be taking going forward. We will continue to work on better model systems for disease. I think this is lacking. Many of the model systems that people have used for studying different neurodegenerative diseases may have been yielding very different structures, as for example I've shown you for the heparin-induced tau filament formation in vitro. We are working on these systems in vitro, but we're also starting to do these things in cell cultures and see if we can make cellular models of disease. And ultimately, we hope that information that we gain from these experiments can then be used to make more relevant animal models, where at least the structures that you see of the tau or, for example, alpha-synuclein filaments in the animal models are at least the same as we see in human. Because as far as we've looked so far for tau, that is probably not the case with currently popular used models. So that brings me to my acknowledgement slide. As I said in the beginning, I'm very grateful to all past and present members of my group and also the group of Michelle Goddard, with whom we do all the amyloid work in very close collaboration. I'm grateful for people at Thermo Fisher, for in particular Abhay Kotecha, for all his work on the prototype machines that we get access to there. And I'm very grateful for all the personnel of the LMB facilities, the EM facility, the computing facility, for all the work they do to be able to for us to do our experiments. And then on the right is a list of collaborators which all give us access either directly to brain material or to filaments purified from brain material, and without that all our work on the amyloids would not be possible. And ultimately we thank the families of those patients who die with these diseases and make their brains available for science for that donation too. And I thank you very much for your attention. I'd be happy to take some questions.
Hello, thank you so much, Sjors, for the invigorating talk and the insightful discussion on your research. We really learned a lot on about cryo-EM, how to, use recombinant DNA technology, or, like protein engineering, or a little bit of standardization of purification to get different proteins, deleting a part of the protein to make it soluble and more manageable and purify and get the structures at a higher resolution. So I think the audience and the participants have learned a lot from this talk. So let me tell you briefly. So I would like to thank you so much once again. I would now like to tell everyone briefly about the award that Dr. Sjors has received. Dr. Sjors is the recipient of the 2022 AstraZeneca Award. This award is generously supported by AstraZeneca. It's made every 3 years in recognition of outstanding work, which through biomedical advances leads to the development of a new reagent or method. The research should have been carried out in a UK or Republic of Ireland laboratory. So we can now welcome questions for Dr. Sjors, and you can type in the questions in the text box so that we can move ahead with the question and answer session. And I have a few as well. So one question, Sjors, first of all, it was a great talk. Can you hear me?
Yes.
Okay. One question I have, that you have two different types of filaments, right? That you were talking about. One is the straight filament and the other is the paired helical filaments. And these two have two different types of structures with a little bit of different conformational organizations, right? So, and you also said that the paired helical filament, you found it a little bit more than the straight filaments. So what is the structural signature that determines that it will form this filament versus that filament? That is one question. And how do you look into the step, and why would this one be more than that? Is it because of the stability of one filament over the other? Have you looked into that aspect as well?
Yeah, so I think those are very essential questions to which we basically do not have an answer yet. So we do these experiments in extractions from brain tissue at the end stage of disease, and we do tend to see more pH depth than straights. Why that is, we don't really know. You know, it could be the amount of the
Can you look into the stability
density,
of these
the
two?
molecules that form the density that keeps the straight filaments together is limiting, but I don't know if we can't really find out why this is if we don't have a good model system to make these. So
Or if you
PHS
make
we
some
can now make,
yeah.
but straight we still can't. So once we can also make straight filament, then you could start to do experiments where you make both at the same time and perhaps in different types of cells and see what happens. But I think, yeah, those are important questions. Why you form different types of filaments in the same disease, or even these completely different structures in the different diseases, is all completely unknown.
Okay, I have another question, is that you showed that you have taken the brain tissue samples of several females having Alzheimer's, right? So you had taken with one, and then you had repeated with few other
Yes,
patient
some of them
samples.
were male too,
So
yes.
yeah, most of them, you have shown that whatever I've seen in that slide, it might be representative, that most of them are sporadic, only one was familial. So is there any difference between these two types of, patient samples in terms of the structure of these filaments, or the tau protein, or whatever structural studies you have done? Any difference
So
in the?
we did not observe differences between the sporadic and the familial cases of Alzheimer's disease in their tau structures.
Okay, so that's interesting. Okay, so that means it's more like, it's more like disease specific, not type specific, probably. Okay, thank you so much. And another thing I wanted to ask you is that you did de novo atomic modeling of these two types of these filaments. I'm going back to the filaments several times. So is it that you had reconstructed these filaments from the cryo-EM structure, or it is like using the sequence you had made these filaments? How did you go about it? If I just wanted to know how this atomic modeling was done.
So the result of the experiment is
Mm-hmm.
the blue and the green kind of three-dimensional density maps that I showed. So this you can load into a 3D graphics display, and then you can build three-dimensional atomic models in them, very much like you would do so in X-ray crystallography. So you have a three-dimensional density map in which you then,
Okay.
but you know the information about the sequence of the protein you're looking at, so that can help you. And then it's looking at the shape of the side chain densities in the map that allows you to dock in this protein sequence.
Okay. All right. Thank you so much. I have a few questions from the audience. One is tau protein tau just 1 second, something got disappeared. Somebody asked, is tau is kind of a prion protein. So that was one question by somebody called Dash, but I'm not finding, maybe they deleted that question. Anyways, I'm going to the next question. It
Do you want me
says
to
that, thank
answer it?
you. Yeah, you can answer it. Yes, certainly
So,
you can answer.
I mean, prion protein, there's a big difference between tau and a prion protein. You know, prions, even if you eat prion protein, for example, you can get prion disease. So that's not known for tau to happen. It's Alzheimer's disease is not contagious. So in that sense, it's different. But we think that some of the underlying mechanism of how tau spreads, tau aggregation spreads throughout the brain are prion-like, in that they also are seeded assembly where these amyloid filaments play a role.
Okay. So there is one question by Dash. He said, thank you, Dr. Sjors. I learned a lot from your great presentation. Do you explore new in vitro models, salt incorporated ones with small molecule ligands, such as the work you did for APN 1607 within tau filaments. So that is a question.
So what the work they refer to is that we used a PET ligand. So you can use PET ligands to do the visualization of tau aggregation in the brain of living patients. So PET ligands are small molecule compounds that bind specifically to the filaments, and then you add a radioisotope and you see it in the PET scanner. So what we did there was we co-incubated brain-derived filaments from Alzheimer's disease brains with a PET ligand, and we did the structure showing where the PET ligand binds to the filaments. So now that we can make the same filaments in vitro, we can do a cleaner experiment. That's correct. We could use the in vitro derived PHFs and add the PET ligands, and we may be able to visualize lower occupancy binding, because as you do these preps from brain, they're always much more dirty than when you do the preps in vitro.
Okay. All right. Thank you. Thank you so much, Sjors, for the answers you have provided. And since there are no more questions, are there? No, there are no more questions. So let us thank you once more. It was an excellent talk. We learned a lot from whatever you have said. It was an excellent presentation. And congratulations once more for the great achievement. And so we thank everyone for attending, as well as we thank the speaker. If you wish, you can continue the conversation online by following at Biochemical Society or at PP Publishing on Twitter as well. We welcome suggestions for future topics, as I told you earlier, to feature in this Biochemistry Focus webinar series. If you have an idea for a webinar in 2022, we invite you to submit a proposal. We can find more information about our webinars on our website. And also you can find the previous recordings. If you even missed some of the webinars, you can go back to the website or the YouTube channel, and you can listen to the recordings from today's as well as past webinars that have been, have been held earlier. Finally, I would like to highlight that in this challenging times, it's more important than ever to stay connected and engage with your fellow molecular bioscientists. It's an extraordinary time for us all, but it's also an exciting time to join the Biochemical Society's community of researchers and specialists to stay connected and take advantage of the key benefits, including discounted registration fees for society courses and meetings, exclusive access to a wide range of grants and bursaries. Please go to our website. I would really suggest you do that. You can have personal and online access to 2 of their journals and more. Visit our website to find out more, because Biochemical Society and Portland Press have lots to offer for your scientific growth. And I again thank all of you for your patience, and Dr. Sjors and the Biochemical Society for this excellent webinar. And thank you all. Thanks for listening. Bye.
Thank you very much.